So, good evening, everyone. Just a moment. It looks like our audio is fixed up here. I'm going to go ahead and share my screen with just a couple of introductory slides this evening. Hopefully everyone can see that properly. I just want to say hello and welcome to our next in our series of our community lecture series. We're going to hear tonight from Dr. Magdalene Seiler and Andrew Browne, who I'll be introducing in a moment. They're going to talk to us about stem cell-derived retinal organoids to repair vision, which has been a really long effort on Dr. Seiler's part. Of course, we want to start out with a few thanks. Certainly to our speakers, but also to our organizing committee, who includes Judy Beck and Brian Cummings, our community outreach chair, as well as UCI Media, without which we could not possibly get anything useful done. And it's Kyle Good who's here hosting us this evening. Quickly, I just want to point you to a couple of things that are up on our website. The 2021 year in review is still there. There's lots of great stuff about exciting things that happened at the center last year. We're super proud of those things and all of the efforts that we're making in order to build upon the recently released CIRM funding, including our newly funded training grants, which received the highest score of all the applications submitted in California. Something else I thought I would just brag about for a minute tonight is some new instrumentation that was installed in the fall. You can read that story from our newsletter, but also up on the UCI news site. We have installed a Helios Hyperion system that was acquired with a combination of gift funds from the Cullen Endowment, and using that to leverage against a large NIH application, which is super exciting. Without the help of Craig Walsh, Matt Inlay, Vanessa Scarfone, and Pauline Wen, who you see in this picture, it wouldn't have been possible to do that. But this is some really unique instrumentation that lets us look 3D at tissue sections and look for proteins and how they relate to one another. It's the only one of these instruments that is at a UC campus currently, and we've just set it up and launched a core facility. So we're super excited there. Oftentimes people ask, how can you help? Well, the number one thing you can do is to participate. Thanks to all of you for attending this evening. We're really grateful to have you participate in our community lectures. Sign up for our newsletter, follow us on Facebook, visit our websites. Any of those things would be wonderful. If in addition to that, you might be interested, we have Giving Day coming up, and please make a gift or think about making a gift. You can contact Amber Harness, who's our development officer. And this is really critical for things like the instrumentation I just talked about. It's the Cullen gift that enabled that to happen. Without it, we would not have been able to launch that new initiative in the center. A couple of words on format this evening. We are, as we always do in our virtual format, speaker presentations will be followed by time for questions. So Dr. Seiler and Browne will go first. And if you put any questions into the Q&A box, we'll answer those if we can on the side as they come in where possible. Otherwise, we'll be holding them and I'll compile them for a moderated question and answer session at the end. So we will get to as many of those as we possibly can tonight. With that, just some brief background on Dr. Seiler, who received her education actually in Munich, Germany, and here at UCI is an associate professor in residence in physical medicine and rehabilitation, and also in the Department of Ophthalmology. Our second speaker is Dr. Andrew Browne, who received his MD from the University of Cincinnati, also a PhD from University of Cincinnati, and at UCI is a clinical assistant professor in ophthalmology associated with the Gavin Herbert Eye Institute, which, as most of you know, is right across our parking lot, a stone's throw away. So with that, I'm going to thank you for your attention and invite Magdalene to please go ahead and share your screen to go ahead and get started with the lecture. Magdalene, you need to unmute.
Okay. Thanks everybody for attending this presentation, and I want to especially thank the organizers for inviting me and Dr. Browne to this presentation. And so you will hear today stem cell-derived retinal organoids to repair vision. This funding wouldn't be possible without the support from National Eye Institute, CERN, and research to prevent blindness, and is based on some patents. And there are many different collaborators that have contributed to this research. And it takes a village to perform research. So I want to thank to all the staff and students that have contributed over the years, and especially Dr. Armand, with whom I've been working with for decades. And over here is the current staff of the Seiler lab. I want now to give the word to Dr. Andrew Browne, who will introduce into the eye and retinal organoids.
Thank you, Magdalene. Nice to have the opportunity to come and speak in this forum this evening. I'm a retina surgeon and specialist over at the Gavin Herbert Eye Institute, and I've spent the last 4.5 years of my time at UCI working very closely with Dr. Seiler on this retinal organoid project, which she bore many, many years before I was even finished with my training. But we found some really nice opportunities to collaborate. When we think about our vision, this photograph here, which I've modified, is intended to show you how our vision is structured. In the central part of our vision, we have very clear high acuity vision. This is how we can read signs on the freeway. But the peripheral vision is more blurry and is more responsible for things like night vision and being able to perceive movement. If you overlay a red circle in the center, this is part of the retina inside the eye that is the part of the retina that's responsible for the very high acuity vision is called the macula. And when an eye doctor looks inside the eye, we think about the cross-section of the eye here, and light comes into the eye and is focused onto the retina, which is this orange-yellow wallpaper inside the eye. And we can take these very beautiful photographs of the retina, as you see on the right-hand side here. If we zoom in on this photograph, we can see that there's an optic nerve, which is where all the information from the entire retina dives into a single nerve bundle and goes back to the brain. In fact, there's about 1.2 to 1.5 million nerve fibers that go from the retina to the brain along this 1.5 millimeter diameter optic nerve. This little area over here is about 150 microns, 0.15 millimeters. And this is called the fovea. This is where we have our highest acuity vision. And this is where we actually have 50% of all of our visual information arising in this small, tiny little area. This is a photograph of a normal, healthy retina. So we see here the macula, as I showed in that previous picture, and the other parts of the retina are considered the periphery. So the central vision versus the peripheral vision. Again, when we look at this cross-sectional view of the retina, we can zoom in on the back of the eye where we have the optic nerve and the macula. And we can see that there in this cartoon are multiple layers to the retina. We'll zoom in a little bit further. But basically, we have this retina sitting on top of a foundation of cells that allow the retina to take light information and turn it into electricity. To make this happen, a very rich blood supply continuously supplies nutrients and takes away toxins from the retina so that it can continuously be able to see light. Amazingly, in this cross-sectional cartoon, we actually have technologies in clinic now that allow us to see the true cross-section of the retina in a patient in a matter of seconds. And this is called optical coherence tomography. So here we have this 150 micron, 0.15 millimeter fovea, where the light comes in, passes through the retina, and it passes through the retina at all locations, but the highest acuity vision, the sharp vision, is right here. The light passes through the retina, it touches this bright line underneath the retina, and it's at this bright line where light is turned into electricity. And that electricity goes back in the opposite direction and passes back towards this optic nerve. So this amazing technology that we can use in clinic allows us to see all the layers of the retina. In fact, if we were to take the retina, cut it in half and put it onto a pathology slide, we would be able to see individual cell layers. And you can see how these layers on a piece of dead retina line up perfectly with this type of image. We can simplify this retinal structure a little bit further, and we can define the inner retina as being blue and the outer retina as being green. And when we see over here, there are multiple layers of cells that make up these different blue, green, and magenta layers. If we think about these individual layers, there's a lot of diseases that compromise the photoreceptors at the bottom of the retina and the retinal pigmented epithelium, a single layer of cells that supports the photoreceptors. In a normal, healthy eye, this is how the photograph looks. This is how this optical coherence tomography image looks. But in a disease like macular degeneration, you can see the profound difference between this part of the retina where the photoreceptors are, as well as the retinal pigmented epithelium are. You lose these cells, you lose vision permanently. So there are a number of blinding diseases that kill photoreceptors and these pigmented cells. The most common ones that I encounter every single day that I'm in clinic include things like age-related macular degeneration. This typically affects people between 60 and until the end of their life if they develop it. And we can see in this picture, there's some little speckled yellow spots and brown spots. And if we take a cross-section through this retina, we can see these big bumps underneath the retina, very different from the normal appearance that I showed you previously. In even more advanced forms of age-related macular degeneration, we see that we have this area of a scar in the central retina where the high acuity vision was, and we see that there's total loss of the cells that used to support vision in the central vision. So these age-related macular degeneration is a condition that typically affects the aging eye. But there are diseases that blind people in youth, things like retinitis pigmentosa. And this is a patient of mine who's 18 years old. And if you compare the cross-sectional image over here of a normal retina with that of my patient, you can see that they've had a natural progression of death of their photoreceptors over just 18 years. And so they've permanently lost vision. And the natural course of this disease is for loss of peripheral vision and then eventually loss of central vision. The eye can go from being 20/20 at the beginning of life with normal vision to even no light perception at the end of life, or even in the 30s and 40s. Everyone progresses at a different rate. So we would really like to be able to help people. Well, we don't want people to progress, but we also want to have solutions for people who have permanently lost vision. So coming back to this cross-sectional image, and if we talk about this cartoon of the inner retina being blue and the outer retina being green, and we look at the individual layers of the cells, we see the photoreceptors in the bottom here and the other cells on the inside of the retina over here. And if we pull them away from everything else and we roll them into a ball, this is what we call a retinal organoid, where you have photoreceptors on the outside and other cells on the inside of the organoid. And this is what our research is all about. So how do we make a retinal organoid? In the last 15 years or so, stem cell technologies have exploded. We have multiple ways of inducing stem cells to become retinal tissues. There's a way of taking a whole bunch of stem cells, having them cluster and aggregate to grow into a ball, and then we physically excise a portion of this ball to make a retinal organoid. That takes about 15 to 20 days. Another way is to take these clusters of cells, put them onto a petri dish, and let them gestate for about the same period of time. And then we see little areas that look promising that we cut out and make retinal organoids. If you remember, the gestation for a human is about 9 months. So the production of a retinal organoid is on the same time course as a human's retinal gestation. The development of the eye in a uterus or in a petri dish is very similar in time course. So we take these organoids, and from 45 days approximately, all the way out to 120 days, we can monitor the progression of these organoids. In fact, we even take them out to over 200 days. So this is keeping small balls of cells alive for 7, 8, 9 months. We have very sophisticated microscopy tools, microscopes that can take pictures at very high resolution. In fact, here is a picture of a retinal organoid that is 46 days old. And each of these little black spots is a nucleus inside a cell. We can see by 90 days or 3 months, the structure has changed a lot. And even by 5 months, the structure has become layered, just like the retinal tissue that you saw in the previous images. So this very advanced imaging technique allows us to non-invasively, in other words, we don't damage the tissue, study these organoids as they develop. And we can even look at the metabolism of the tissue. We can know what type of metabolism is occurring at any place within the tissue. This microscopy technique is called two-photon imaging. And Dr. Seiler and I use it frequently and throughout our research collaboration over the last 4 years. In fact, there's a host of techniques for looking at retinal organoids. You can take a photograph of them under a standard microscope. You can kill the organoid and stain it like a pathologist would, and you can see the layers of this tissue. And then this is the two-photon imaging, where we don't kill the tissue, and we can look at very high resolution at the structure as well as the function. So in this picture here, we see vitamin A derivatives. Vitamin A is critical for vision, and its derivatives are constantly cycled in the photoreceptors and allow light to be turned into electricity. Here we again see this metabolism, where we see this unique metabolism in each of the different layers of these retinal organoids. And if we zoom in really close to the surface of a retinal organoid, here's the organoid. You can see these little buttons coming off the top of it. These are the photoreceptor outer segments. These are the areas where the photoreceptors actually can turn light into electricity. And if you look at a human retina under the microscope, you can see these outer segments here as well. So these organoids do develop into having these mature photoreceptors. So the question is, how can we manufacture these organoids in a reliable and reproducible fashion to be able to use them as a medical therapy or simply to use them as a research tool? These organoids can be used eventually to hopefully restore vision or to understand how vision loss occurs. So when we make these organoids, just like an M&M factory, we can have a lot of outliers. And this requires a lot of manual labor, a lot of man hours, continuously feeding the cells every 2 days, replacing all the nutrients. And it's a lot of human error that can be introduced and a lot of variability. So we want to take something that looks like this and make a production line that's manually independent, can be automated, can reduce error, and can reduce variability of the organoids. So we all are eating M&Ms that are as we would expect them to be, and we can all receive organoids that are as we would expect them to be. So can we improve manufacturing? Well, we can. The conventional method for maintaining organoids in a petri dish requires a person going and replacing all the media, this fluid bath that surrounds the tissues every 2 days. And this happens throughout the week. And each time they go and do this, this can take several hours. Well, then the question is, can we put these little organoids into a tiny little chamber and flow fluid through that chamber and exchange media, give new nutrients, take away toxins in a continuous and automated fashion? And so to do this, we use a variety of tools that have become very popular for hobbyists, but also can be used in advanced science. We use laser cutters, 3D printers, which are the two on the right here, and I have these tools in my lab. These tools were supported with CERM funds to be able to have them available so that we could make a device to continuously and autonomously maintain organoids over months. So here's an example of a high-resolution image of a mold, just like a Jello mold, that we printed on a 3D printer. This is about 2 millimeters in diameter here. And the height of this little area over here from the base to the top is about 200 microns, 0.2 millimeters. And we can make these silicone devices where we can flow media in, feed an organoid, and flow the media out. In fact, we can scale this up quite a lot. And so we can have a continuous single line to feed organoids continuously for many months. So the way that we do this is we have a media reservoir, and we have a simple syringe pump, a pump that connects to a syringe and pulls the syringe plunger back, and therefore continuously pulls media through a bioreactor. So here is some bright field microscopy, some simple microscopy images of organoids over time, 41 days after they were born, all the way up to 158 days. And you can see how they get larger, and you can eventually see these photoreceptors on the surface. These are organoids that were maintained in a conventional petri dish. And these are organoids that were maintained in one of our bioreactors. And structurally, they look very, very similar. So we feel very confident that we can put organoids into the bioreactor and maintain and receive organoids that look almost identical to the organoids that were made in a petri dish. But what's the big difference? Weekly human labor and variability. The bioreactor takes about 10 minutes every week for a human, whereas the conventional culture takes 3 to 4 hours per week for a human. And this lasts, 4 to 9 months. So it's a huge labor cost over time. So then we use our very fancy 2-photon functional imaging to look at the organoids and compare them between bioreactor and a static culture. And this is the high resolution image, just a structural image of the organoids produced in both. We can look at metabolism between the two different types of organoids. And we can see that there's maybe more of a signal here than in the organoids maintained in a conventional petri dish. And if we look at the actual content of the metabolism, is it glycolytic? Is it oxidative phosphorylation? It's really not different, but the total amount is clearly very different just from looking at this image. And if we look at oxidative stress, we can see the bioreactor organoids, there's less red than there is in the static organoids. And the red is an indicator of oxidative stress, where lipids that make up every single cell in the organoid become oxidized. And so we can image this non-invasively, and there's a very clear, statistically significant difference between organoids that are continuously receiving fresh food versus organoids that receive fresh food every 2 days. So there seems to be a major benefit to perfusing organoids continuously in a labor-free bioreactor. So the bioreactor does improve manufacturing. It takes out a lot of human error and reduces manual labor. I'm going to have this just brief conversation about stem cells and their use in medical practice. Just because it says stem cell technology doesn't mean it's good or ready for people. In the earlier part of the last decade, there was a clinic in Florida that performed liposuction to pull up fat and obtain blood from peripheral veins and put it into a process where they separated out cells. They then injected those cells into people's eyes who had macular degeneration. This was without any prior randomized, controlled, placebo-controlled studies where there was support for doing this procedure. There were 3 patients. They had 20/60 and 20/30 vision. So this eye was good enough to drive. The next patient was 20/50 and 2100, not good enough to drive. The third patient was 20/40 and 2200. The 20/40 eye is good enough to drive. And after the injection, the first patient had no light perception in either eye. They could not tell whether the lights were on in the room. The other patients had a very similar course. This was after they received medical care to treat severe, profound inflammatory complications from a medically unproven procedure. So stem cells are very exciting. I have great optimism for their potential in the future, but there are risky situations where patients who are vulnerable, for example, losing vision, might try anything that they can possibly imagine to try and restore their vision. But the only way to do this safely is if the medical technology and intervention has been studied in a randomized, controlled, and very heavily monitored situation. And we're hoping to get to that point in the near future. So at this point, we can make retinal tissues in a petri dish. How well do they work at restoring vision? And so I'll turn it over to Dr. Seiler. I'll stop sharing my slides, and she will continue with some of her slides.
Okay, so we are using to test this question, how do transplants can transplants affect vision? We use rats as an experimental model, and we use the same instrumentation that Dr. Browne talked about, optical coherence tomography, to look at the eye of the rats, of live rats. So here you have an example of a retina of a healthy rat, here of a rat that has lost its photoreceptors, and here is an example of a rat where we have put a transplant into the subretinal space. So, but we want to transplant human tissue into rats, and for that we need to use rats that have a defect in their immune system, so that human tissue grafts can survive without having to use drugs, because the drugs would interfere, would make the rat very sick. So these are called special nude retinal degenerate rats. So we start out with a ball, the retinal organoid. So we need to transplant a sheet. So how do we do that? We have to cut pieces from it. And so this is an example of retinal organoid pieces, and they have an exact orientation because the photoreceptors are on the outside. So we have to put it into the nozzle of the delivery instrument in exact the same way. So we keep control of it. And in the rats, we have to put we have to use go through the back of the eye to reach the subretinal space. So this is different than human surgery. So we will have the tissue in the nozzle, and this shows the transplantation instrument that can place it in the subretinal space. And as I mentioned before, we use optical coherence tomography to monitor the transplant over time in live rats. So this is a transplant early after transplantation, and this is much later. And so it has developed different structures we call rosettes. And we can also see how the transplant is placed and how well it survives. Now, how do can we measure the rat's vision? So the rat cannot tell us the letters on the eye chart. So we have to put the rats into a box with moving black and white lines. And if the rat can see the moving lines, it will turn its head in the direction of the moving lines. And these lines have a different width, so it's much easier to see these thicker lines than the thinner lines. And so the rat is moving back and forth. And this is an example of our data comparing transplants with controls, so sham surgeries and no surgery controls. And you can see that the transplanted eyes consistently maintain better vision than the controls up to 6 months post-surgery. The way to test the vision is to measure electrical activity in the rat's brain. When we place a flash in front of the rat's eye. So we map the whole surface of this brain structure called superior colliculus. And if you do this with a non-surgery retinal degenerate rat, we don't get any responses. And the same thing with sham surgery, where we haven't put a transplant in. However, when we put a retinal organoid transplant in there, we can see responses in a specific area of the brain. Maybe it's not perfect, it's not covering the whole surface, but it's we cannot not find these responses in the controls. And then finally, we use different staining methods to identify the transplant after. So we process the tissue after the end of the experiments, and then we stain the tissue slices to show whether the transplant, where the transplanted cells are and what they do. And so this is an example of the transplant cells labeled in green and photoreceptors labeled in red in the subretinal space. And this is the host retina on top of it. So to sum up, stem cells can be coaxed into making mini retinas, retinal organoids. And retinal organoid transplants improve vision in rat models of retinal degeneration. So what is coming next? So our research is geared towards coming to a clinical application, but before we get there, we have to produce consistently clinical grade organoids, and the safety and efficacy of the transplants needs to be confirmed at a contract research organization. So outside from potential buyers from us, with very consistent protocol. And once we have those data together and a protocol for clinical trials, then that can be submitted to the FDA for approval of clinical trials. And estimate the timeline for this would be maybe 3 to 5 years, depending on how successful we are. Okay, so I want to thank everybody for listening to this, and please ask questions.
So thank you, Magdalene and Andrew, for just really a beautiful pair of talks. So clear and nicely laid out. I really appreciate it. We have a few questions that have come into the Q&A, and I'm sure that there are going to be some more. I invite everyone to please submit your questions through the Q&A. We have plenty of time, which is unusual for us to be able to answer them, so that's terrific. So the first one that I'm just going to highlight for you guys, I'm going to read them out loud so that everyone can hear and follow us, is whether or what techniques and factors are used to induce stem cells in order to get them to take on this retinal phenotype that you're searching for. Either one of you.
Magdalene, I'll defer to you for that. It's
okay. So there one has studied the retinal development, so there are people using specific factor, but there's also another protocol that just is based on spontaneous differentiation and not so complicated. So either methods work. They're just each lab has kind of different approach on how to do this.
So are there any, I'll just interject a question. I haven't seen this asked yet. It's a very long differentiation process. And so as you know, but maybe our audience doesn't entirely know, the longer the differentiation process, not only the more complicated in terms of the sorts of issues that you all are trying to address for time and consistency for production of cells, but it's an opportunity for contamination, for chromosome abnormalities to arise, for all kinds of things that happen that make it more complicated under GMP or good manufacturing practice protocols to be able to bring this into the clinic. So is there any way to, I'm sure that you and others have been looking at it, is there any way to shorten that process, or is that a complication that because of the bioreactors becomes, you know, something that you can overcome and it's not so meaningful?
So actually for transplantation, we use younger tissue. So we don't go for 6 months. We go for, so for most of our papers, we have used tissue that was around 2 months old, but it may be that 3 months is a better time point. But there are other researchers that trying to coax stem cells directly into becoming photoreceptors, but then we are kind of missing the 3D approach. And the other retinal cells are important. So people have genetically modified stem cells to kind of get organoids that only produce these cone photoreceptors that are important for high acuity vision, and also to reduce the number of the inner retinal cells in the organoid, so that the percentage of photoreceptors in the organoid is much higher.
Great. So another question from Sid Golub, who both of you know well, is whether vitamin supplements that include things like lutein which are often recommended to older patients to slow the progression of macular degeneration, are those supplements necessary for the growth of retinal organoids, or do they affect the growth of retinal organoids, and what's the consequence for those in terms of retinal cell function? I think he probably means in vivo there.
Yeah, it's a great question. I'll answer the first part of it in terms of a clinical component of these vitamins that are used in diseases like macular degeneration. Macular degeneration was studied in a large cohort of patients in the earlier part of the century, and they continue to be followed. And in this cohort of patients, people either received a vitamin cocktail of vitamin C, E, copper, zinc, lutein, and zeaxanthin, or placebo. And they showed that the rate of macular degeneration progression was reduced, especially for people who had intermediate to advanced disease. So this cocktail of vitamins has demonstrated to be beneficial in the context of age-related macular degeneration. It hasn't been demonstrated to be of use in other blinding diseases like retinitis pigmentosa or Stargardt's disease, or even myopic degeneration. However, there were a number of studies back in the 70s and 80s looking at vitamin A supplements for people with retinitis pigmentosa, and the data was very weak if there was any signal at all to be seen. And so it isn't routinely recommended to take vitamin A supplementation. It's really up to the clinician's kind of desire. In terms of studying the host of vitamin supplements in retinal organoid development, I'm not aware of research that has looked at them specifically, largely because these supplements are intended to reduce oxidative stress at the end of retinal life when there's a disease process, whereas when we're developing organoids, it's the very beginning of life. It's the in utero phase of retinal development. And certainly retinoic acid, which is a form of vitamin A, is important to the differentiation of retinal organoids. So vitamin A still remains very important to manufacturing retinal organoids. Some people do and some people don't use it. Some people use taurine, some people don't use it. So the reality about retinal organoid science and its use in studying the biology of retinal development and the eventual use in therapy is that we have many people around the world that each have their favorite recipe, and they all make their fruitcake a different way. And so there isn't a standardized protocol. So that's what we hope to do with something like a bioreactor, is we could say, okay, we'll standardize all the conditions that are mechanical and human in nature. And then we can just have a whole bunch of different types of Kool-Aid that we pour on the organoids and eventually fine tune the process.
Yeah, that's a great way of phrasing it. I think really making it accessible. That's a great description. Magdalene, I'm going to turn the next question to you because I'm compiling a couple together from the audience. I hope for those in the audience that I'm capturing this well in a compilation. And it's really about mechanism. So one question is at what stage the organs are transplanted, as you said, 2 or 3 months after organoid differentiation, after induction. At that stage, how laminated is the structure, the organoids that you're transplanting? And do you think that the mechanism of how they're giving repair has to do with making new connections, with making new synapses between the transplant and the host or the recipients?
Yeah, so at that stage, the retina has more or less two layers. So an outer layer and an inner layer. And at that time point, we have the retinal ganglion cells in the organoid. But with longer term culture, so the ganglion cells that would project to the optic nerve, they die off. So the inner part of the organoid actually deteriorates the longer time you culture it. And one reason why I would like to move from 2 months of age of transplantation to 3 months or even later age is that so far most of these transplants have formed so-called rosettes, so that's are rolled up photoreceptors, which is not the nice parallel layers as we have in normal retina. But we have indications that the transplants make synapses with the host retina. So we have specific markers for the host cells, and we combine that with markers for these synaptic connections, and we can look at this in high magnification. So it definitively plays a factor that there is a connectivity between transplant and host, and these transplants, they really connect very nicely, integrate. Yeah.
So I'm just going to take the liberty of asking a follow-up to that, because I think it will help people in the audience understand a little bit. One of the things that's complicated about cell-based therapeutics is that their mechanisms of action can be what we call pleiotropic. So in other words, kind of multifactorial. It might be that they're making synapses and improving the environment for all of the other recipient cells that are around them. Do you think that that is a factor likely here, or do you think it will be possible with this strategy of transplantation to really say there's a single mechanism of action in the case of the retinal organoids?
That might be difficult. So we cannot completely exclude that there was a trophic effect of the transplant. So one way of future research would be to try to identify factors that the tissue can produce and kind of compare it. So see what extracts of the secretions of those cells can do in a retinal degenerate host. But I would think you need active live cells for this. You couldn't really just inject something, because that would then be depleted, and then the degeneration would continue.
Yeah, exactly. And so it can be that the cells that are transplanted have a role as sort of a factory that's going on over time. And I just want to highlight, you sort of said it, Magdalene, but to bring out again for the audience, that while this is a complicated nature of cell therapeutics, and it makes it a challenging one in terms of the FDA authorization process for a clinical trial, it's also probably a benefit, right? That these cells, by virtue of being transplanted, are most likely doing more than one thing that could yield some benefit. So next question from Nate Kenner, and I'm going to adapt this a little bit. So I think this is referring to a study where you're transplanting cells into your rodent model of macular degeneration. How long did those studies continue on? So what was your last time point of assessment? How long did it take to get some improvements in visual function?
So almost some of them almost 1 year. So we are kind of limited with our rat housing costs. So normally we design to end the experiment with these recordings from the brain, because that's a terminal experiment. But we have some few where we went longer time. So 10 months and 10 months is also possible. So we are also, another limitation is the health of the host animal. So because we are using rats that have a defective immune system, the older they get, the more there's a chance that they get infected by something, and then they just can turn sick without any reason. And all the work is down the drain. So that can that can happen.
It's a challenging animal model.
Yeah.
So many of us in the Stem Cell Research Center share that conundrum. I think, Andrew, the next couple of questions clearly fall into your courts. And this is, what's the potential if this kind of retinal organoid transplant were to prove to have some efficacy in terms of your initial clinical trial target? What's the potential, do you think, that might be able to transfer to other conditions? So myopic macular degeneration is called out, Stargardt's disease. So any comments on that?
Yeah, so with specifically retinal organoids, we're aiming to replace damaged photoreceptors and some of the cells that connect the photoreceptors to the optic nerve. There's a potential, and Dr. Seiler's been doing research on this for some time, to transplant organoids with pigmented cells as well, the RPE, as a co-graft, two different tissues made at the same time and then put together like a peanut butter and jelly sandwich and then transplanted. In that case, it opens the opportunity for retinal organoids to be able to help people who have lost their RPE as well. Myopic macular degeneration is a devastating condition that if it really takes a hold of Asian countries, blinding people in the working age. It's a structural disease of the eye. For a transplantable tissue to function, you have to have the surrounding tissues be normal as well. In one of my slides, I showed that we have the layers of the retina, the pigmented epithelium, and then a rich blood supply underneath that. And that blood supply is critical for the organoids and the pigmented cells to live. Unfortunately, in advanced myopic macular degeneration, we actually lose the blood supply as well. So whereas conditions like retinitis pigmentosa, Stargardt's, even age-related macular degeneration still have a normal blood supply, they can probably more likely benefit from this type of technology. There are hosts of other hereditary retinal diseases where the blood supply is normal, or even the pigmented cells are normal. So it all depends on what is the milieu or the composition of the tissue once it's generated to the point that it has.
Yep, and that's a great, really comprehensive answer. I'm going to skip down a couple of questions and then come back for our audience because, a subsequent set asked about, potential applications for partial retinal artery occlusion or for glaucoma. And again, I think, Andrew, what you're saying is these would fall into the category of things where there's the structure around may not be fully intact. And so there might be only so much a strategy like this could do in terms of repair. Yeah,
for the retinal organoid flavor of stem cell transplantation based technology, right. Other diseases like glaucoma and retinal artery occlusion, vascular occlusions, which are basically strokes, those will benefit from the technologies being worked on many other scientists around the world.
Yeah, and I think really critical to recognize there is the kind of work that the two of you are doing, which is bringing together the biology of stem cells, how to achieve this sort of differentiation, development of techniques for bioreactors, and combining multiple tissues together. These are all things that are piecemeal, right? But they have spin-off applications. They have implications for how we understand other aspects of a host of different eye diseases. And so immediate application, no, but that's the thing about science, right? You're always learning something, and this data comes together to help across the board.
Yeah, I think one thing that we haven't really mentioned is that you can take cells from patients with inherited diseases and then make stem cells out of it, induce pluripotent stem cells, and then make organoids. So the issue is, of course, that this is still very immature tissue, but one can study some mechanism of metabolic problems and other issues that happen with the photoreceptors, and then one can try to do gene therapy, by example, or use different drugs to improve the situation. So have these organoids as a kind of drug testing factory in vitro.
Right, which is a really critical application, like screening applications for stem cells. I think that kind of leads us into well, maybe not exactly. It doesn't lead us into that as a follow-up question. So I'm going to turn the tables on you and ask something slightly different. Dr. Richard Robertson, who's a former UCI faculty member, is on tonight, and he asks about the capacity of stem cells. Well, it does sort of lead into this. So about stem cells to be able to turn into any mature stem cell type, right? And this is really what you were talking about, Magdalene, that you can take a patient's own cells and reprogram them, take them down the path of making retinal tissues or other tissues. And so his question is, there must be specific growth factors that push towards retinal cells. Are these like well known? Are they published? Are they out there? Can any lab pick up on the sorts of findings that you and others working on parallel projects in the field have? Or is this a secretive industry and nobody shares?
There are lots of publications. So it was actually already in like 2007 or earlier, people published about retinal organoids, and the techniques have been improved, but it also and there are many groups working on this, and but probably the very nifty details of the procedure, you still have to be highly trained to be able to do that, because you need to recognize how a good a developing organoid looks like, and distinguish that from any other potential tissue that can develop from stem cells. Like if brain tissue develops, that's not what we want to have, and that would behave very different.
Yeah. So one of the things,
and that's
so there are very clear criteria. Very clear tests that determine that we are on the right, that one is on the right track to make a retinal organoid, specific genes, specific markers, etc. Yeah.
a lot of the studies have a common thread. You know, some of the specific factors that are commonly used are Wnt inhibitors and ROCK inhibitors. But like I mentioned earlier, everyone has a favorite recipe, and they have a lot of variability. Magdalene mentioned that making the organoids is a very skilled process. And so if you want to make a custom organoid for someone to transplant into their eye into the future, you're going to have a highly trained person to be able to identify that what is a good and a bad organoid. So one of the goals with a bioreactor is not only to control the delivery of growth factors, nutrients, take away toxins, the simple stuff, but is to develop an artificial intelligence technology surrounding simple microscopy to identify what is an organoid that's turning into a piece of brain versus an organoid that's turning into a piece of eye. And, you know, for the host of organoid technologies that are made for pancreases and gastrointestinal tracts and all these other model systems, we have the technology to do these things. It's just a case of really barreling down and having the time to do it. So I'm very optimistic that we will have standardized tools to make any type of organoid tissue that will be supported by very simple engineering tools. And although artificial intelligence is not simple, it's become kind of like a standard laboratory tool where we can eliminate the need for highly trained individuals to be able to identify what is a good organoid and allow a standard cell phone to do that for you.
Yep. And that's, I think you bring up a really important issue that's linked, a really important issue that's linked to this, right, is cost of cellular therapeutics, right? And especially as we move towards patient individualized therapies, really that kind of precision medicine aspect. And the critical part is, I'm just going to point out for the audience, what Dr. Seiler and Dr. Browne are working on is that entry-level stage to demonstrate not just feasibility in the lab, but move that through to clinical testing, right? And we certainly are hopeful that this is going to be wildly successful. When it is, being optimistic, when it is, then that changes the landscape entirely, because now getting investment into developing AI to artificial intelligence to be able to do better recognition of when you have a good organoid versus a bad one, when you're down the right differentiation path versus not. That will be a flood of research, additional research to what's already going on. And that changes the landscape in terms of how to deliver therapeutics and what the viability is that from a commercial point of view, from an expense point of view. And so it's step by step, these things follow one another, and you have to get through the steps that you guys are doing right now in order to make that more viable. I do want to address one quick question. I'm sure at this point we're not going to get through all of them, but I'll keep going as long as you guys are willing to stay here. And that is, given that you're predicting 3 to 5 years for approval in the US, I think, let me make a distinction there. What they're looking is 3 to 5 years until they have authorization to do a clinical trial in the US from the FDA. Have you considered jumping to other jurisdictions which might offer a speedier process? And this is quite complicated. So people think the FDA is slow because it's cautious. And I think that's an unfair perception because this is an issue of safety first. So if one were to go to any number of countries that doesn't regulate any of their therapeutics, particularly cell therapeutics, as closely as the US does, I would make the case, and I'm going to ask you guys to comment, that you're much more likely to end up with something like the situation that Dr. Browne described in Florida in this clinic that escaped regulation than you are with a well-conducted clinical trial that's going to give you real efficacy data that's really going to move the field forward and lead to real treatments. And so that's the challenging thing to understand. It looks easy to jump outside the US, but it's a little bit penny-wise, maybe a lot of it penny-wise, pound foolish. So Andrew in particular, would you like to chime in on that?
Yeah, I think we all want to go to the moon, and we want to get there and bring people back, and we want to go to Mars, we want to bring people back. It's a similar kind of idea. We have to do it in a very thoughtful, prospective way, where we think of everything possible before we start doing it with people. And whenever are we going to be the first people to put organoids into the eye? Maybe not. Are we going to be the first people to put organoids into the eye in a very controlled and safe environment? Maybe. Very, very likely. So I wouldn't be surprised if there are studies going on around the world where people are receiving organoid-like tissues in a transplanted fashion where it's not regulated. I'm not sure how rigorously it will be studied. Maybe it'll be just fine, but I wouldn't recommend any of my patients to it.
Right. Great answer. Magdalene, next one to you, which is a complicated one, but I think an important one to ask. And that's why rats and not a closer relative to human, like say a non-human primate?
Yeah, to use a non-human primate, there's first of all, there is an ethical problem. They're not closer to humans, so you have to jump through much more hoops, and also you wouldn't be able to get enough number of experimental animals to really do statistics about transplant effects. And the rats are relatively cheap, and you need to, in order to really be sure that your experiment works, you have to repeat it and repeat it and repeat it. And if you can then really say that the outcome is repeatable and significantly better, you need to have enough numbers. And if you wanted to use primates or pigs, then there are no immunodeficient animals in this, and then you have to treat them with drugs. And we had done also a treatment of immunocompetent rats with drugs, and then the experiment, it gets much more difficult because the drugs also make the rat really sick, whereas these immunodeficient rats, they're just fine as long as they're not hit by a virus infection.
And just to expand on what Dr. Seiler was explaining, when the retinal organoids that you guys are working with, which are human-derived, go into humans, that's what we call an allograft, an allogeneic transplant. And you can control that relatively well. That's what all organ transplants are with immunosuppressive drugs. When you take human cells and put them in a normal immunocompetent rat or in a non-human primate or in a pig model, that's what we call a xenograft, a xenogenetic transplant. And there, the immunological barrier is much, much higher. The rejection is more aggressive. It's not just that they're difficult experiments to do, it's difficult to get enough cells to survive to really be able to assess the kind of endpoints that you are. Right. So I guess this one can go to either one of you. In terms of the transplantation paradigm that you have, is there a potential for things like biomaterials, hydrogels, or other sorts of biomaterials that are out there to actually help you in terms of integration? Is that something that you've thought about as a path for your research?
I'm thinking about that in the context of combining the retinal organoid with retinal pigment epithelium, because these are actually two separate tissues. The retinal pigment epithelium requires very different culture conditions than the retinal organoid. So we have to make a sandwich just before transplantation, and to put them together, we have to use some kind of matrix. So there are other researchers that are actually trying to use a special matrix to combine specific cells like the photoreceptors and the RPE in this matrix with very orienting that orients the photoreceptors, because if you dissociate them, they're kind of hard, but to keep them in this oriented status and let them grow like in a normal retina. So there's a lot of research going on, especially from people that want to transplant dissociated cells, because it's better, the survival is better if you put them back into a matrix, transplant them as a sheet, than if you just squirt cells into the eye. They tend to die. So
yeah, they like to have a structure, like a scaffold to hang on to, and that may help the tissues that are already within the eye as well. So great question. So I think this is kind of an interesting one, even though we already talked about synapse formation. But the question is, if the transplant alters the photoreceptor signal, right, or the transmission to the brain, how like the brain takes time to decode that. How does that work, right? So is it the same amount of time as a normal intact animal who doesn't have macular degeneration? Is there a longer delay, but the signal still gets there in your transplants? Do you have any information you can share on that?
Yeah, so the principle is that the transplant connects, there's still some remaining inner retina. So the signal to the brain comes through the host retinal ganglion cells. So that circuit is the same. However, the time of the response time to the signal is a bit longer in transplanted rats compared to normal. And that's probably because there's some kind of havoc with the synaptic connectivity. There might be some extra cells in between, so it takes longer time.
Yep, great answer. That's exactly what I was looking for. So 2 questions, and then I think that we will wrap up. The first one is, I think, a clarification for the black and white moving bar experiment. I forget the name of the behavioral device that you use for that, which is assessing basically, well, so I guess what I want you to do is to comment on the kinds of questions that you can ask rats to give answers to in terms of behavior, right? So the question here is, if you use color bars, will that tell you about color vision? And the trouble is, we can't get rats really to tell us so much about color. So we're asking them to tell you about what they can detect in a different way. So maybe just explain that experiment one more round. What are you asking a rat to tell you?
Yeah, so it's called optokinetic reflex. I didn't want to use that word, and it's essentially it's a reflex. The rat has to do it if you can see the moving stripes, and one. Another thing besides thickness and thinness of those stripes, one can also investigate whether the rat can see the contrast, because here we have a high contrast black and white, but you can make that black more gray, so that the difference to the white is much less, and that way you can study contrast sensitivity. And I haven't seen anybody trying to use this for color vision. Would actually be interesting if we could study this, because the human photoreceptors, they can see more red colors, whereas the rat can only see green, blue, and UV. So the spectrum of what the rat can see is different than what the human can see. So that might be something for the future to test that out.
Well, great. I didn't know our audience was going to give you scientific ideas today. That's awesome.
Yeah.
The other thing is the human macula is a very unique thing. Primates have a similar idea of being able to see very precise colors, but rodents, while they do have cones and rods, cones being the color vision, the number of cones and the spectrum of cones, the lights the different colors that they can respond to, is much narrower than in humans. So certainly one could investigate whether a cone-rich organoid would improve cone color vision, but it's in the animal model itself. It's not something that animal sees the same way as a human.
Right. Exactly. So the questions you can ask them have some scope. Magdalene, go ahead.
So I mean, one could then just like in the recording, instead of white light, present some color to the rat. But it's a little bit difficult, because normally we use red light to prepare the rat, so then the red light is already saturated away to see responses from rat's cones. So we would need to change the experiment, maybe work with infrared for the surgery, and then use red light.
All right. So last question, I think we have a good wrap-up spot, which is probably Magdalene to you, but Andrew, I think there's some clinical implications. There's an underlying question here. And so that is, what might be some of the side effects of retinal organoid implants? So for the rats after surgery, was their health impacted in any way? How long do you follow up? What other sorts of things do you see in those animals, if anything? Of course, the parallel question here is what might one expect when you finally move to clinical trials? So maybe Magdalene start, and then Andrew wrap us up for the evening.
Yeah, so the surgery in the rat is, of course, much more difficult than a human eye. It's a smaller eye, and so sometimes what can happen is that we induce some damage, and that damage we can see then in the optical coherence tomography, and we can eliminate those rats. But there are things that can go wrong with the surgery. But otherwise, once the transplant is stable, there's actually nothing that can happen with it, provided the organoid source is really a retinal organoid and does not contain any stem cells that would proliferate.
Yeah, and then as far as once this sort of technology is ready for human trials, anytime that an organ is transplanted, you can develop an inflammatory reaction, an organ rejection. And the eye is a very unique environment, just like the brain and actually the testicles. It has a privileged environment, meaning the immune system can't get to it very easily, but a surgery is going to make that a little bit easier. In the patients who were in Florida, they didn't necessarily get an infection from the cells being injected into their eye. They experienced a vicious and blinding inflammatory rejection of the material that was put inside their eyes. And that inflammatory reaction actually caused blood vessels not to flow anymore. And so they developed no blood flow in their eye. So do I think that's going to happen in humans? Not necessarily, especially if we're able to do it in a controlled and monitored way with the potential for using single or multiple immunosuppressive drugs. And eventually with customized, tailorized organoids that are made from the same source as the human, then that organ rejection becomes much less serious. But the benefit is that we truly have a wealth of knowledge that we can benefit from our renal, hepatic, and other organ transplants, cardiac transplants, in terms of managing systemic inflammatory conditions. And that can be beneficial in the eye as well.
Yep. And of course, there's parallel research that's ongoing that is looking at ways to make these kinds of allogeneic transplants, human into human, sort of immuno-invisible, right? As a path to be able to get stable engraftment and stable benefits and minimize the risk of rejection without having to necessarily be on chronic immunosuppressants. That's a ways off, but it's certainly something that stem cell research field is aspiring to. So with that, I think we should call it at 8:10. I'm sure everyone joins me in thanking Dr. Seiler and Dr. Browne for just really a great set of presentations, so clear, and what a lovely job that you guys did. So thanks for joining us tonight. And my thanks to the audience for being with us again for this series, and we look forward to seeing you next time.
Thank you for the opportunity.
Thank you all so much for your interest. Hope to see you soon.
Bye-bye.
Bye-bye.